So this is the fourth video on cell cycle course. In this video, we will learn about MPF regulation. So in the last video, we have learned how three independent experimental approaches led to the discovery of structure of MPF. So MPF is made up of one regulatory element known as cyclin B and one catalytic unit known as CDK, or this is a cyclin dependent protein kinase. Together they are known as the MPF. Now in this video we will learn about the MPF regulation. So throughout the G2 and S phase, the cyclin B and the CDK1, they keeps on associating and forming this couple. However, in this position, they are not in the active form. With the help of V protein, phosphorylation takes place at threonine 161. This phosphorylation is necessary for activation of this CDK. However, at the same time, phosphorylation takes place at two other positions, threonine 14 and tyrosine 15. These two phosphorylation makes this CDK inactive, although it has got the phosphorylating phosphorylation at threonine 161 and the cyclin B. Later on, the dephosphorylation of these two positions leads to activation of the MPF. When the MPF becomes active, it phosphorylates a number of proteins in chain, which ultimately leads to progression into mitosis. At the same time, it also triggers the degradation of cyclin B by proteolysis. So this proteolytic degradation of cyclin B brings about inactivation of the CDK, and so it exists the cell cycle. It exists the mitosis, it progresses into the cytokinesis. And again in S phase and G2 phase, it will form association with cyclin B, and this runs in a cyclic manner. So there are different families of cyclins and cyclin dependent kinases. For example, CDK1 and cyclin B are members of the families of related proteins. And different members of these families control the progression through different phases of the cell cycles. For example, CDK1 controls G2 to M transition in association with B type cyclins. Just the thing we read just now. CDK is working with cyclin B. Similar CDK, the same CDK, this controls passage through start and entry into the mitosis in association with G1 cyclin and CLNs. So, cyclins, different members of cyclins, they work with different cyclin dependent kinases. So, with this, we come to the end of this session. And we have learned about the mechanism of CDK regulation, which takes place at four levels. One is association of cyclin with CDK. Two is activation at threonine 161, which is activating phosphorylation, which is necessary for the activity. Three is inhibitory phosphorylation at threonine 14 and tyrosine 15 positions. Apart from that, there is another level of regulation, the fourth mechanism, which is association of CDK inhibitors. So when CDK inhibitors gets associated with this MPF, it will not work, it will be deactivated. So thank you.